Hi, and thank you for joining us for Chardan's Virtual Ophthalmology Summit. The focus of this installment is on rare ophthalmic diseases. My name is Daniil Gataulin. I'm one of the senior biotech analysts here at Chardan. It is my pleasure to introduce Rob Lynn, CEO of Avista Therapeutics, and Leah Byrne, Chief Scientific Officer. Rob, Leah, nice to meet you. Thank you for joining us today.
Thank you for having us, Daniil.
Yes, so the session that will follow for this summit is going to be a fireside chat format, and I look forward to discussing Avista and the programs. So with that, let's go ahead and get started. If you guys can spend a few minutes to introduce yourself and provide a brief overview of Avista and its programs.
Fantastic. My name is Rob Lynn. I'm the CEO of Avista Therapeutics. My background is that I'm a recovering scientist. I did a PhD in genetics at Harvard and have experiences in corporate and business development at Millennium Pharmaceuticals and the Bill and Melinda Gates Foundation. First came upon the project that became Avista when I was a VC with UPMC Enterprises and funded the Series Seed round and loves the company so much that came on board as CEO a couple of years ago. Leah, you want to do a quick introduction?
Absolutely. I'm Leah. I'm the CSO of Avista Therapeutics, and I'm also an assistant professor at the University of Pittsburgh in the Department of Ophthalmology. I did my PhD work in gene therapy for retinal disease, and I've been working on this on this problem since my PhD years. And yeah, now serving as the CSO of Avista.
Excellent. Thank you. So let's jump right in and talk about the platform that's at the heart of your company, the scavenger platform. Now it enables you to identify promising candidates for your lead program, potentially other future indications. So first, let's talk about this platform. What is it? What does it allow you to do?
Maybe, Daniil, if we take a step back from Scavenger, tell you about our thoughts on Avista and why the company is necessary and why we're providing something that I think will be helpful. I think the unfortunate news over the past week here on the deaths in the Sarepta gene therapy program and trials really highlights the need for better tools. And I think that's where Avista is focused on creating better tools for the delivery of genetic medicines. And our feeling is that by creating more effective vectors, we can lower the doses. And what we've seen both in the eye and systemically is that there's a dose-dependent issue on terms of the safety concerns. And so as Leah mentioned, her graduate student work is actually probably the beginning of the thread of Avista. She and her colleagues at Berkeley discovered AV2.7M8, which is the first intravitreally delivered engineered gene therapy vector. And it works really, really well in mice where it was designed. So there should be no blind mice, no matter what the fairy tales tell you. But in NHPs and humans, we think that the effectiveness of that is a little bit less effective. And so when Leah and I first met in 2017, when I was at UPMC Enterprises, I asked her, what do you want to do next? And she said, create much better versions of 7M8. And of course, being very inquisitive VC, I asked, first of all, why and how? And I think the why is summed up in sort of the effectiveness of tools like 7M8 as you graduate from mice to higher organisms. And then how is the scavenger platform that you had mentioned previously. But in 2021, we I led the Series Seed round of $10 million. In 2022, we signed a partnership with Roche on using our technology to think about vectors. And I think where we are today is that using the platform, which Leah will be happy to tell you about, we've actually created a best-in-class intravitreal vector that we think is between 1 and 2 logs better than 7M8 in terms of effectiveness and efficiency, which will allow for hopefully dose reduction and a reduction in those inflammatory responses through intravitreal injection. But even beyond that, we have been able to take a lot of the data that's been generated in the early years of Avista and use that to design some machine learning vector libraries that we're calling Avista prediction libraries that we think will actually even create even better vectors than what we have right now. And so really thinking about enabling that intravitreal route for better delivery of genetic medicines beyond what we have currently in our pipeline, but also thinking about how we can use scavenger and machine learning in other tissues and other indications beyond ophthalmology, which we'd be happy to talk to other folks about partnering. So I think that's sort of the nutshell, and maybe you can continue with the question on scavenger.
Yeah, actually, if you can pause here for one more second, see if there, obviously, 7M8 has been widely recognized as quite efficient. Improving upon that is even more impressive. Besides the increased transduction efficiency, what are some of the potential other limitations of the established vectors like 7M8 or others that are being used?
Leah, you want to take that?
Yeah, absolutely. So yeah, as Rob mentioned, part of my PhD and postdoc work was developing 7M8 back at UC Berkeley. And 7M8 was designed in the context of the mouse retina and was developed through a process called directed evolution, which we were obviously very excited to create 7M8, which has now become sort of the gold standard for intravitreal gene delivery. But because it was developed in the context of mouse, it's not optimal for overcoming some of the structural barriers and obstacles that exist in large animal eyes. And those are barriers that are shared between humans and non-human primates, which is a model that's very important to work in order to create therapies that are really going to translate into patients. So another difficulty with 7M8 is, or the approach that was used to create 7M8, is that it can be very slow. It can be a very time-consuming, expensive process because when we created 7M8, we were really focusing on photoreceptor cells. And the way that we did that screen was relying on an animal model that had a GFP, a fluorescent green reporter, in those photoreceptor cells. As I mentioned, it's really important to work in non-human primates in order to create therapies that are going to translate into humans, but we don't have monkey models that have fluorescent green reporters. And so, a lot of the other vectors that have been created in mice, they're just not able to translate really efficiently into humans, and the process that we use to create those vectors really doesn't work in primates. And so that really led us to the creation of the scavenger workflow. Our goals in creating scavenger were really to create something that was going to be highly quantitative, really reliable, really result in vectors that were going to make an impact on patients. And really importantly, we wanted something that could be done efficiently, directly in a non-human primate, and allow us to look at specific cell types in the non-human primate. And that's the workflow that we developed. And as Rob said, we've been able to leverage this pipeline to create best-in-class intravitreal gene therapy vectors.
And so,
got it. Okay.
Sorry. Well, one additional clarification. I think in addition to being better at transducing cells in 7M8, 7M8 has a tendency to be focused in the foveal region, and I think with our vectors, we're actually getting transduction in the mid periphery and the far periphery, as well as the central foveal region. And so I think that's another advantage that we've been able to engineer into our vectors.
Okay, got it. Understand. So let's go. Maybe if you could walk us step-by-step process of actually arriving to that vector with those desired properties. Maybe from all the way from creating that library to selecting the best candidates.
Absolutely, yeah. So the way the scavenger workflow works is that we first create a library of AAV variants. So these are maybe 10 on the order of 10 to the 5th or 10 to the 6th different variants, unique variants with a different capsid structure. And each one of these is packaged with a unique genetic barcode that allows us to track the performance of each one of the variants in the pool. And we are able to create all kinds of different libraries, including insertion libraries, where we put amino acid inserts into positions on the capsid that we know are important for receptor binding. We can make libraries based on a lot of different serotypes of AAV. As Rob mentioned, now we're really incorporating more and more machine learning into the design of the libraries that we test and put into the scavenger workflow. But again, once we've created these variants, they each get this genetic barcode that allows us to track the performance of each one. They all get pooled together and then injected into a tissue of interest. So, of course, right now we're very focused on intravitreal gene delivery in the eye. So they all get injected into intravitreally injected into non-human primate eyes. And so after injection, each one of the variants in the pool has an opportunity to transduce cells in the retina. And some of the variants in the pool are going to be unsuccessful. So because of the change in the capsid structure, they're not going to be able to infect cells. But some of the variants will have improved properties. And so after each of the variants has had a chance to transduce cells, then we take the tissue out of the eye and we create a single cell suspension. So we're looking at individual cells now from the tissue. And we run those cells through a single cell RNA-seq pipeline that allows us to look at the transcriptome profile of each one of millions of cells all in parallel. And so by doing that transcriptome analysis, we can do two things. First, we can identify the cell type of each one of the cells that we're looking at. So we can identify if it's a rod cell or a cone cell or a retinal ganglion cell based on its profile. And then we can also quantify the performance of each one of the variants in the pool based on the presence or absence of those genetic barcodes. And so this is a really powerful approach to very quantitatively analyzing the performance of a very diverse pool of AAVs. And this allows us to have single cell resolution on AAV transduction profiles. And it also allows us to evaluate the AAVs based on RNA expression, rather than just if an AAV is able to attach to a cell or get inside of a cell. Our requirement is that the AAV actually produces mRNA. So it's a little bit of a higher bar to meet than other screening strategies have previously applied. And so at the end of this process, we get a ranking of how well each of the variants in the pool has performed. And very importantly, all the variants are tested in the same non-human primate, along with control vectors. And so that overcomes sort of another barrier that's existed in the field. Non-human primates are they're not inbred like mice are, and so they are very variable. Much like humans, each monkey is very different from the next monkey. And so it can be very difficult to accurately compare vectors when you inject one in one monkey and another in another monkey. But with the scavenger workflow, everything is done head-to-head in the same animal. And so it's a very quantitative and reliable approach for us to evaluate all the vectors in the pool.
Okay, got it. And a few clarifying questions here. First, is that a reiterative process? Once you identify, a candidate that performs better than your reference, but still not up to your internal standard, do you use that candidate to create a new library and potentially identify or continue to identify vectors that are better and better until it hits your threshold?
It is iterative in the sense that, yeah, we have created this really reliable data set that, again, was created directly in vivo in non-human primates. And it's that data set that we're relying on, that we're sort of feeding into our machine learning pipeline now. And that is resulting in our second or third generation of capsids that Rob mentioned earlier.
Okay. And then what do you say are the main limitations of this approach?
The main limitations of the scavenger workflow, I would say, honestly, this has overcome a lot of the barriers that we're really trying to overcome. And I would say that
yeah, I think the sequence space that we're looking at is really broad. So if you think about the insertions we're doing currently looking right now is it's a 6 amino acid insertion. So there's 20 to the 6th power in terms of number of different combinations. And so even in the library, when we're looking at millions at a time, it's tough to cover all that space. And so I think we've got points within a sequence space. And I think that's where the machine learning that our dry lab team has been really involved in helps to both cut down on that sequence space, but also identify some of the potential amino acids that might be important in that. And so allows us, as Leah mentioned, sort of an iterative process to design better libraries on the next screen.
That's an excellent answer, and I definitely agree. Yeah.
Great. Yeah. Thank you. And so given that this results, or you working on identifying the best vector for intravitreal administration, and intravitreal, of course, has a slightly higher risk or higher risk of inflammation that can compare it to subretinal or even suprachoroidal space potentially. What fraction of the population would you say would be expected to be neutralizing antibody positive, and is that a concern?
We do not anticipate that being a concern. So there are two things. One, intravitreal delivery is still, it's very local delivery. It's not a systemic delivery where I think those NAb titers would be more of a concern. And we're also, through our workflow, we've designed vectors that are on the order of a log, maybe 2 logs, more efficient than previous vectors. And so that allows us to reduce the dosage of the vectors significantly. So you did mention that intravitreal delivery is a little bit more immunogenic than other routes of delivery, like subretinal injection. But yeah, we really feel that with these more efficient vectors, we're going to be able to deliver gene therapies below the limit that's going to stimulate a significant immune response. So that's really critical, that we're creating much more efficient AAVs that are going to allow us to reduce the dosage significantly.
Okay, and then in terms of the precedent for a prophylactic regimen, you could still, I would imagine, rely on previously designed prophylactic regimen, including the one that's used by 7M8s, correct?
Yes, we'd anticipate doing something sort of similar to other trials. We wouldn't, but again, we feel like we're going to be able to reduce dosage significantly and would anticipate less inflammation than in other clinical trials that have required higher dosages.
Gotcha. Okay. And also, as you're making these, how are you testing for manufacturability, and is there a potential risk that your best candidate is going to be the most challenging to manufacture?
Yeah, that's a really important question. So obviously, manufacturability is an important issue. Less critical in ophthalmology than perhaps in other, areas where really high amounts of AAV are required, but still it's a very important issue for all gene therapies. So we've incorporated a step that ensures manufacturability into the workflow. So we always are focusing in on vectors that we know are going to be able to package efficiently. And as we've incorporated more and more machine learning into our approach, that's been one of the gates that we've incorporated into our workflow to ensure that that we're able to manufacture the AAVs, but they're also strong packagers too.
Got it. Okay. And so with this approach, and there being quite a big number of potential indications in ophthalmology, what else are you looking at? What are the other possibilities? What else are you thinking in terms of expanding?
In terms of indications?
Mm-hmm.
So I would say that probably any indication could benefit from a more efficient AAV vector. I think that's really a theme that's emerging from the field right now, is that efficient gene delivery is still really key, really critical, no matter what indication you're going after and no matter what route of administration you're using. And so I would argue that almost any indication could benefit from improved AAV vectors.
And so, Daniil, we have an internal pipeline that our lead candidate is currently in XLRS, but we're also looking at additional ones in inherited retinal diseases. Roche, as I mentioned, we have a partnership with, is working on their own indications. But I think, as Leah said, we think that the universality of the improved intravitreal vector is something that can be used across a range of indications, probably many more than we can do ourselves. So we're happy to discuss partnering with other folks who want more efficient vectors.
Got it. Excellent. So let's switch gears and talk about that later indication. So XLRS, X-linked retinoschisis. First, if you can spend maybe a couple of minutes on the disease background, what it is, who gets it, how common it is, and what is the current unmet need?
Absolutely. So XLRS is X-linked retinoschisis. This is a disease caused by mutations in the RS1 gene, which encodes for a protein called retinoschisin. Retinoschisin is a secreted protein that's made by photoreceptors and also bipolar cells. And this protein is really important for the structural integrity of the retina. It's important for adhesion between retinal cells and also important for organization of the retina. When there are mutations in the retinoschisin gene, what happens is that the layers of the retina end up splitting apart and these cystic cavities form in the retina. So this is an X-linked disease. It affects young boys. They're typically diagnosed when they fail vision exams in school. And as a result of this disease, they lose visual acuity and are more prone to retinal detachments over time. So typically the disease is diagnosed when the boys are young, and it can be fairly stable for a few decades of their life, and then can progress further, usually in their 40s or 50s. That being said, it is still a pretty heterogeneous disease, and so it can be different from patient to patient. Currently, there are no approved therapies for XLRS other than some carbonic anhydrase inhibitors, things like that, and just diagnosis and trying to be careful not to get some sort of retinal detachment. So this really does represent a significant unmet need. It's one of the more common retinal degenerations, monogenic retinal degenerations in children.
Okay, got it. So as you mentioned, the disease begins manifestation in childhood, right? But then it may be fairly stable for a number of years, if not several decades, which potentially represents a very broad therapeutic window, right? Or the treatment potential window. Within that, would you still try to target patients at a younger age? Would you expect there to be a correlation between when that treatment is given and the potential effect?
So that's something that I think we really want to answer with our upcoming through clinical trials. That's really the only way to answer that question. Yeah, we'll be interested to find out the answer.
Got it. And in terms of I guess different stages. So obviously when it first diagnosed at early stages, then as it slowly progresses, what's potential population of those patients would your therapy be able to target?
Maybe I'll take a whack at this answer, Daniil, but I think we commissioned a LEK study to look at the commercial population for this. And so the prevalence appears to be around 25 to 30,000. If you look at the US and the EU population there, roughly split about 50-50. And the market that we can address, I think, really is going to depend on the exact question you posed, which we don't know the answer to right now, but is how long can the layers of the retina be separated? And then when you put them back together, they still actually function normally. And I think that is something where we think, obviously, if the younger the patients are, the less time the retinal layers have been separated. and probably more effective there. We don't know if there's a limit or a timing there, but that will be sort of the effect on that prevalence population that we can address. But we really do believe that with an intravitreal therapy, as opposed to a subretinal therapy, that we can address a large percentage of those that will have their vision improved by an XLRS gene therapy. So I think that's one of the things that we really believe. And when we look at the commercial analysis that was done, is that both the patients and the providers are a lot more comfortable with an intravitreally delivered therapeutic, especially in the younger population and in the population where, as you mentioned, there's some stability in that vision. I think there's a reticence to do a subretinal surgery on those folks.
Got it. Okay, makes sense. And so as you are gearing up to submit your IND, right, potentially this year, right?
I think we're gonna run into, as usual, some optimistic projections at the beginning of the year. So I think we're pushing that into 2026. But we do have preclinical both safety and efficacy studies that are underway in progress, and really trying to have those lead to the IND filing, as you mentioned, but now probably in 2026.
Okay, got it. In any case, yeah, that's not that far away. And as you are approaching this milestone, can you highlight some of the key data, that preclinical data that you've collected today that give you the confidence in the program?
Yeah, so I think in terms of efficacy, we're studying the effects of our therapeutic in knockout mice, and I think deserve to give some credit to one of our scientific co-founders, Dr. Paul Sieving, for the work that he did on XLRS at NIH, really pioneering this work and showing that there are knockout models of XLRS that you can rescue in mice, both structurally and functionally. And I think that's what we're hoping to reproduce or improve upon with our therapeutic. And then other companies in the field have also followed that pathway of doing efficacy studies in the knockout mice, and then some of the safety studies in non-human primates. And so I think we're following along a path that Dr. Sieving initiated, but other folks have done to get to an IND here.
Got it. Okay. And as you're thinking about designing the trial and the endpoints, obviously the early one is going to be focused on safety, but for it being a rare disease, you'll also be collecting some efficacy data as well. When you look at efficacy, what early endpoints would you say would be critical to show improvements on?
Yeah, so fantastic question. I think structurally, we'd love to be able to see some of the schisis closure through the intravitreal delivery route in the subretinal delivery route from other companies. They've been able to show some schisis closure, probably less in the areas that are outside of the blebs, but I think that is a really important structural endpoint that we're looking for. But at the end of the day, I think when we talk to providers and particularly insurance folks, that they really want to see functional recovery for these patients, and whether that's BCVA, microperimetry improvements, or even low light vision, trying to figure out at the end of the day, does the patient experience improve? Does their vision improve via our gene therapy? And I think that's the real goal at the end of the day.
Got it. Understand. So you mentioned, right, the competitors program. They use a subretinal approach. They do have some innovative capsids as well, right? The laterally spreading capsids that technically allows them to travel away from the bleb. What are some of the key learnings that you've observed and took away for your company from the early clinical trial of Atsena?
Yeah, maybe Leah, you can comment a little bit on the capsid and how that is, and then I'll give some of my thoughts about the early clinical data here.
Sure. Yeah. Yeah, of course, we've watched this clinical trial with interest. And I think one of the things that really distinguishes our approach from what it's seen as doing is the intravitreal versus subretinal injection. And I would say that even with a spreading vector, the fact that a subretinal injection is still required in XLRS, I think, does present some challenges. So one thing that at Avista, we really want to be sure to do is to be able to treat the spectrum of patients that exist. So patients that have schises in the periphery, for example, or patients who might have significant schises in the center that would make it difficult to perform a subretinal injection in that area. So yeah, I think that's really what distinguishes the two approaches, even with the spreading capsid.
And I was going to say, I think that leads to some of limitations in terms of commercial uptake for that type of strategy, where you have to find where the schises are and map them out and be able to plot where the blebs are close enough to affect vision in the fovea, but don't interfere with the present schises. And as Leah mentioned, subretinal delivery is still a pretty specialized surgery. But I think what we do see from at Sienna that we're taking with really great heart here is the flexibility that the FDA has shown in the recent approval of combining their phase 1/2 with a pivotal trial that allows them to skip that end of phase 2 meeting to plan the pivotal. And presumably they've agreed on some endpoints there that would be approvable. And I think that's a big step forward for the field, and hopefully something that we can draft into as we think about our regulatory strategy as well.
Got it. Makes sense. Yes. Yeah, I think it's important for the company who's not or who is closely behind to have that precedent set and the more established route for you to follow. Excellent. So a few more questions before we conclude the session. How are you thinking about your CMC strategy and where you are in terms of manufacturing the product for clinical trials?
Yeah, so we're pursuing an outsource strategy with a CDMO. We're working with Forge Biologics over in Columbus, Ohio, and I think early on the capsid has been performing very well, and I think we have a platform for both clinical supply, but also an eye towards eventual commercial supply. So I think we've been very happy with our relationship with Forge, and also really happy with the progress on the CMC front. So I think our CMC person, I say when he looks bored, that's good news for the rest of us. It's when he looks panicked that it's bad news. And so he's been looking pretty bored recently. So I think things are going well.
Got it. Excellent. Also, you mentioned about Roche partnership. Can you tell us about that, how important it is for Avista, and if you have any updates on any potential targets or candidate selections?
Yeah, so we signed a partnership with Roche very early on before we had any capsids to show them. So they bought into the platform, similarly to what we did on the VC side is buy into the platform at a very early stage. And so really happy with their support early on, and especially the non-dilutive capital that that relationship was able to provide us. We can say that they have taken a capsid that we have discovered into their program, and last time we spoke to them, they seem very happy with the progress of both their programs and the capsid internally. One good thing in agreement is that they're doing their own manufacturing internally, and have reported again very clean and very good CMC news there as well. So I think they're excited and happy about moving forward. Unfortunately, I can't comment on any progress in their pipeline. We'll leave it to them when they announce their progress in their gene therapies and using our capsid. But I think the partnership has gone very well, and we're excited to see what they do with our capsids.
Excellent, excellent. And in terms of the funding, can you give us a highlight of how much you've raised to date and what milestones are you funded through?
Yeah, so we raised a Series seed round of 10 million. We're looking for about a $50 million Series A. UPMC Enterprises committed to a co-lead of that round, looking for another potential co-lead to help with that. I think that would give us runway until a data readout on XLRS in the phase 1/2, where we're able to get a safety readout there and hopefully a hint or even further efficacy signal. Really excited about moving that forward. But as Leah mentioned earlier, and Daniil, as you asked earlier, where there are a number of different indications where we think our vectors can really improve gene therapies. And so we're looking forward to talking to other partners about licensing out our vectors to their programs as well.
Got it. Excellent. And I guess last question is, what do you think investors are still potentially missing about Avista's story?
Yeah, I think for all the clinical and commercial hurdles in gene therapy, I think both Leah and I, and I'd say speak for everybody in our company, we still think genetic medicine delivery is really important, and the promise is still there to be able to treat a large number of patients, and we just have to be able to recognize that perhaps better delivery tools are needed. And I think we've got a real good head start on some ones in for retinal delivery, and we think that we've got potentially even better ones on the way. But thinking also about how we use Leah's platform scavenger combined with the machine learning that we've already done on the capsids in the eye, thinking about expansion to other tissues as well. We know we can't do everything. So whether it's additional partnerships in ophthalmology or tissues beyond where better delivery vectors are needed, we're happy to talk to folks and investors as well about broadening the platform beyond what we've done already.
Got it. Excellent. Well, on that note, I think I'm going to thank you again for joining us. It's been great having you, and really appreciate the discussion. Thanks, Rob. Thanks, Leah.
Thank you very much for having us.